Platelets. The last lecture on blood, and then we'll be done with the second unit. So what is a platelet? A platelet is like the dandruff of a bigger cell called a megakaryocyte. So it's a fragment of a megakaryocyte. So basically you have this huge cell out here, and it sheds its cytoplasm, and these are the platelets. Okay. So platelets are fragments of megakaryocytes, shedding of a megakaryocyte. They circulate in the blood for about 9 to 12 days. About 9 to 12 days. Okay. And then they're eventually removed from circulation by the spleen. Okay, so we've seen there are two structures of the blood that are removed by the spleen. Okay, and also remember red blood cells. Red blood cells will also be removed by the spleen as well. Okay. So what do they do? What is their function? Okay, so we know that the red blood cell is responsible for carrying oxygen and carbon dioxide. We know the white blood cell is responsible for inflammation and infection, okay, or the inflammatory response. And so the platelet is responsible for initiation of the clotting cascade. Okay, clotting cascade. So the process of making clots. Okay, it is responsible for patching holes in the vasculature. Okay, response to that, and to maintain of hemostasis. Okay, now so hemostasis has got a word that actually means what the word doesn't mean. So if you remember, stasis means to stop, and hemo is blood. So a lot of people, when they look at that, they're like, oh, you're going to stop the flow of blood. Hemostasis in this instance means the stop of loss of blood, or to allow proper blood flow. So like, let's just say that you get in an accident and you cut off your hand gets cut off. You're going to start spurting blood everywhere. So, but basically hemostasis is to prevent that blood from leaving the vasculature and squirting out. So hemostasis will stop that bleeding. Okay, so that's basically what hemostasis is. And whenever you're in the hospital and you say, I need a hemostat, basically you're just needing something to stop bleeding. So now, hemostasis, we're going to spend the rest of the time talking about hemostasis and doing a clot. So hemostasis, like we said, is to stop bleeding, or is to stop some sort of vascular injury. So there are 3 parts to it. And I use the example of like losing a limb or cutting yourself to do this. So let's just say you cut yourself. And you're bleeding. And so there are 3 steps that occur to stop the bleeding. Okay. And so the first step is what's called the vascular phase. Okay, vascular phase. And basically what's happened is called vasospasm. And so what vasospasm does is it contracts the muscle that lines the blood vessels. Okay. And though that's how it works. So to illustrate this out, let's say we have a blood vessel here. We have a blood vessel. Let's get that like this. We have a blood vessel. Underneath this blood vessel, you've got muscle, and underneath that, well, actually underneath the endothelial lining, you've got this connective tissue layer, and this is collagen. And collagen is the fiber of the connective tissue. It is the fiber of the connective tissue. Okay, that's what collagen is. Fiber of the CT. So if you get an injury in here, let's make it a small eraser. So let's say you get an injury right here. And what you do is you have exposed collagen. We're going to make our platelet in green. The platelet's going to bounce along this. It's going to bouncy, bouncy, and then it's going to get caught in there. And so what that's going to do is it's going to signal off, hey, there's an injury in here. We're bleeding, most likely. And so what that's going to do is it's going to cause muscles. There are muscles in larger arteries. Okay, you've got a layer of muscle around here, and it's actually circular. Muscle is circular, and it's going to cause it to contract down and stop the blood flow. It's going to stop the blood flow. Okay, so that's it. That's it, the vascular phase. 2, you get what's called the platelet phase. Okay, and so what happens here is when that platelet, that initial platelet gets stuck in the collagen, it's going to send out a signal saying, hey, I need more platelets. And so what you're going to do is you're going to get a bunch of platelets. They're going to sit and they're just going to bunch around this. They're going to just bunch up around this, and they're going to wall off that injury. Okay, and what this is called, okay, this is called recruitment and adherence. Okay. Recruitment and adherence. Okay, and basically you're going to get an aggregation of all these platelets to the injury. And then 3, you're going to get what's called the coagulation phase. And what this is this is going to be the formation of a clot. Okay, the formation of a blood clot. Now, the platelet phase will make a platelet plug. Okay, it is different than a clot. It is different than a clot. A platelet plug is the best example. Let's just say you have bricks. Okay, you're building a wall of bricks. Okay, and you put your bricks on like this. And, you know, in any given circumstances, this would probably work pretty well. You know, you could have this stand, and, you know, and it wouldn't fall down just on its own, okay? But you wouldn't want to build a house like this. You just wouldn't want to stack bricks to build a house. Okay, problem is something hits it, it's going to cave in. If, like, stiff winds come, it's going to fall. What you need to do is you need to put something in there to stabilize it out. And what you do is when you're building a brick house is you put mortar in here. And what that does is that gets all these bricks to actually stick together. And when you get all of these bricks to stick together, it is pretty strong. It is really, really, really strong. Okay, this is the way platelets are. So if we imagine each one of these bricks is a platelet, the mortar, when you have all these bricks mortared together, that's what a clot is. You can't have a clot without platelets, but without the mortar, it's not a clot. And what this is fibrin. Okay, it is a fibrin clot. Okay, and what the coagulation phase does is it takes fibrin that is dissolved in the blood, gets it to undissolve and fill in and solidify the clot. Because if you don't, the platelets will fall off. They'll eventually fall off because blood is moving incredibly fast through that vessel, and it will take the platelets with it. Okay, so we're going to talk about the coagulation cascade in very general terms next, and that will be the last what we do. Okay, so the clotting cascade. Okay, the clotting cascade. Okay, there are 2 things that can initiate it. So the first thing that can initiate it is this thing called tissue factor. Okay, so the goo that actually comes out of cells when they're injured, okay, or platelet factors, platelet. Okay, and what platelets factors do are is when the platelet binds to the site of injury, it releases these things that start the clotting cascade. Okay, so now what a cascade is. What a cascade is. Let's say you have one element. Let's say this is element A1. Now, when you activate element A1, it's going to activate element A2. When you activate element A2, it's going to activate element A3. When you activate element A3, it's going to activate element A4. And that's going to activate element A5. That's going to activate element A6. That's going to activate element A7. Okay? So you have to have activations of each one of these individual things in order to get the end product. Okay? So that's what a cascade is. You activate one, then you keep doing it. You keep going. You activate it 2, 3, 4, 5, 6, 7, 8, 9, 10, however many it is, until you activate the end product. Okay? This is exactly how the clotting cascade works. Okay? What we want to do is we want to activate fibrin. Okay, that's our goal, is to activate fibrin. Okay, so there are 3 pathways that the clotting cascade can have. So the pathways. Okay, you have this one that's called the extrinsic. You have this one that's called the intrinsic pathway. Okay, and then these both come together to form a common pathway at the end. Okay, the extrinsic pathway is activated by tissue factor. Okay, the intrinsic is platelet factors. Okay, and then eventually what we're going to do is we're going to form to join the common. They'll join the common pathway, and then we will activate fibrin. Okay, so here's how it actually works, and this is the best way I've ever seen it done. This is how I was taught it in medical school, and this is the way that I'm going to teach it to you. Okay, now all of the factors are labeled via Roman numerals. There's Roman numeral 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and 13. 13. Okay, I don't want you to remember them all. It's not important that you remember all. Okay, what we need to do is we need to know that there is an extrinsic pathway over here, and that is dictated by, we're just going to say factor 7. Factor 7 right here. Okay, and then we have the intrinsic factor, intrinsic pathway. Okay, remember this is platelet factors. Let me spell platelet right. Okay, platelet factors. Okay, we will say this is 12. This is factor 12. Okay, so both of these together, they're going to activate other things. They're going to activate other factors, but eventually they're going to get to 10. They're going to go to factor 10. This is called the common. Okay, this is the common pathway. And then factor 10, it's going to activate this thing called thrombin. Okay. And then thrombin is going to activate fibrin. Okay, and then fibrin is going to form the clot. Okay, so that's all I need you to know about this. I need you to know there's an extrinsic, I need you to know there's an intrinsic, I need you to know there's a common pathway. Extrinsic is factor 7, intrinsic is factor 12, and the common factor is factor 10. It is factor 10. Okay, and the ultimate goal of factor 10 in the common pathway is to activate fibrin and that is going to help us stabilize our clot. That's all we need to know. Done and done. Okay. So now that we make a clot, now we know what are some of the things that we will inhibit clots. So inhibitors. Okay. You guys need to know about aspirin. Okay, so aspirin. This inactivates platelets. Okay, aspirin. This is by far probably the most important of them. When somebody's somebody's having a heart attack, what is the first thing you do? Take an aspirin, because heart attacks are caused by clots that are growing too big. So you inhibit the platelets, you're going to inhibit the clots. Okay. Another one that you are well aware of is warfarin. Warfarin. It is a dirty drug, but it's something that you probably see quite often if you take care of your older patients. Okay. This inhibits clotting factors. Okay. So if you inhibit clotting factors, you will inhibit the formation of clots. You know from people who are on warfarin, they have to get tested regularly, because if their clotting levels are too low, they're just going to spontaneously bleed. They'll bleed into the leg, they'll bleed into their skin, they'll bleed into their joints. And it's very bad. It's very, very bad. Okay. Another one that you've seen is heparin. Okay. This is a factor 10 antagonist. Okay, so what that means is it prevents factor 10 from working. So if you prevent factor 10 from working, you can't make new clots. So the one thing that I want you to know about heparin is that if you give somebody heparin who already has clots, you're not doing anything for them. What you're doing is preventing the formation of new clots. Okay, prevents new clots. Okay, I guess the same thing to say for warfarin and aspirin, but heparin, I've seen a lot of medical students I've seen is like, oh, they have clots, give them heparin. I was like, that's not going to do anything. Okay, so those are the big three. Those are the big three. There's other ones. There's protein C and antithrombin 3, but these three right here: aspirin, warfarin, and heparin. These are the ones you're going to see most often. Okay, there are some new drugs out that prevent clots that work better than warfarin, but they're expensive and people don't like to spend money. I mean, insurance companies don't like to spend money spending on them. Okay, so these things that break clots is the last thing we're going to talk about. Break clots. Okay, here it's called TPA, and you will see this TPA. It's tissue plasminogen activator. Okay, this will physically break down the clot. It breaks down fibrin. It breaks down fibrin. Okay, TPA. This is one of the drugs that you can give people who are having heart attacks. If you can't get them to a cath lab, you can give them tissue plasminogen activator and it will break apart a clot and you can save her life. Now, I'm not going to say which one is better. I'm not going to say which one is better because the research is out to still out. But if you can get people with the current guidelines for people who are having a heart attack, if you can get them to a cath lab within 90 minutes, you always get them to a cath lab before. You use TPA if you can't get them to a cath lab within 90 minutes. Okay, the other one is plasmin. It does the same thing, it breaks down fibrin. And then there's a third, streptokinase. That will break down fibrin. So you'll see these, all three of these do that. So that's how we break down clots. And with that, we are officially done with the blood.